So, hello everyone. I would like to welcome you to a new MGBS lecture. And I am so pleased today to have the chance to introduce our guest speaker, Dr. Gayle Volk, plant pathologist from United States Department of Agriculture. Dr. Volk, she has specialized in the conservation of vegetatively propagated crops and crop wild relatives, primarily fruits, in the USDA-ARS National Plant Germplasm System at the National Laboratory for Genetic Resources and Preservation in Colorado since 1999. She identifies strategies to accrue and maintain genetic resources in diverse vegetatively propagated collection and provides decision support tools to evaluate the breadth and depth of collection diversity. She develops widely applicable and cost-effective methods to cryopreserve clonally derived germplasm, such as citrus, grapes, plants, garlic, small fruits, apple, and many others, and then uses these methods to cryopreserve national plant germplasm system collections as dormant buds and shoot tips. Recently, she has been developing cryotherapy methods to eradicate pathogens from plant tissues. Dr. Volk is the chair of the U.S. Apple Crop Germplasm Committee, who authored the globally strategy for the conservation and use of apple genetic resources and the global strategy for citrus conservation. She coordinates and efforts to develop online training resources that will be made freely available for educating students and GenBank employees about plant genetic resources conservation and use. Dr. Volk, thank you so much for joining us today, and please start your presentation.
Okay, thank you very much for this opportunity, and thank you for the nice introduction, Dr. Salam. Today, I will talk about the USDA National Plant Germplasm System. It's our national gene bank, and we'll talk about some planning we're doing for climate change and some efforts to secure our collection so it's safe when climate change happens. And I'll also tell about some outreach activities we're doing. So plant gene banks are present around the world. There's over 1,700 of them, and most of them store seeds that are placed into cold storage, usually at the refrigerator temperatures, but sometimes in freezer temperatures as well. If they're in the freezer, the seeds will usually last longer. This is a secure way to store seed collections. However, every now and then, the seeds need to be regenerated because they either are depleted because they've been shared too many times or because the viability drops. And these are times when the seeds have to go out into the field and they're vulnerable to climatic issues. Sometimes we can't even conserve collections as seeds. And this is an example for many of the crops in the National Plant Germplasm System. I've shown some on the screen, such as garlic, potato, citrus, and date palm. And instead, we keep these in lots of other forms. These could be in tissue culture, they could be in a greenhouse or screen house, or they could be in the field, growing as an orchard or as a planting. These are particularly vulnerable to climatic and pest and pathogen disasters because they are exposed all the time. It's not just during regeneration, but they're always sitting out in the environment or in a building that could be destroyed if a disaster happens. These are kept clonally as plants because they don't make seeds, or because we have male and female plants, such as in date palm, that we want to store. Or maybe the seeds themselves can't be stored easily, or maybe it takes many years to reach reproductive maturity. Or the seeds don't capture the genetic composition of the cultivar, which is the way most of our fruit crops work, because they're grafted rather than propagated by seeds. The USDA National Plant Germplasm System stores both of these types of plants, both the seed-propagated and the clonally propagated materials. We have over 200 different crop collections located throughout the United States at 22 locations. The mandate of the National Plant Germplasm System is really broad. We acquire, we maintain, we regenerate, we secure through backup storage, we distribute, we document, and then we even characterize and evaluate our plant collections. We have over 16,000 species, and we share over 230,000 items each year. With universities and research and breeding programs throughout the world. In fact, about a third of the accessions that we share and distribute are to places outside of the United States. We really see ourselves as a global resource. We have over 600,000 unique accessions within our GenBank. About 566,000 of those are maintained as seeds, which are 93% of the collection. And about 40,000 are clonally propagated, which are about 7% of the collection. So we have a lot of diversity to work with, and that makes it really fun, but it also makes it very challenging. Here's just a map showing the different locations around the country where we have our GenBank located as part of the GenBank system. And the sites that are in red dots on the screen are those that are mostly our fruit crop collections, whereas those in black are mostly seed collections. I personally work a lot with the fruit crop and the vegetatively propagated materials, so I have a lot of relationships with those that are in red. The dot in the center of the screen, I'm not sure if you see my cursor, is Fort Collins, Colorado. We're towards the middle of the country, and that's where I'm located, and we have both seeds and clonal crops here. So we recently published a paper in Crop Science, and that's what I'm going to be talking about today, in part. This paper was called Safeguarding Plant Genetic Resources in the United States During Global Climate Change, and it's free access, so anyone can download it and read it. We also released an ebook, and we've been writing ebooks to share information in a little bit more of a general sense that maybe wouldn't go into a peer-reviewed paper, but people should still know about. And here's a link to our ebook on the left-hand side of the screen, and it's called Conserving and Using Climate-Ready Plant Collections. And in this ebook, we have 7 chapters that talk about plant gene banking and also the impacts of climate change on botanic gardens. We just published, we wrote an application, and this application predicts the climate using a number of climate models at each of the locations of the USDA GenBank. So it's a way that we have a tool for the people at the locations of the GenBank to know what some of the maximum temperatures and rainfall and precipitation and other things that they may be encountering going forward, so they can take that into account as they plan for climate change. Now I'm going to show some examples. These are some examples of real things that are happening in our GenBank right now and reasons why we need to be preparing for future problems that could happen. On the left-hand side, we have some regeneration cages in Prosser, Washington, where they're regenerating alfalfa in the cages. And they have pollinators that they put in there, and that allows for the plants to be regenerated properly without cross-contamination with the other materials that are getting propagated or that are getting regenerated. We do the same thing for sunflowers in Ames, Iowa, when they need to be regenerated. But you can see in this picture a huge disaster that happened in the sunflower regeneration a few years ago when windstorms came through and destroyed the effort. In June 2021, in eastern Washington, which is on the western side of the United States, we had a heat dome, and that area experienced 46-degree temperatures, which we've never seen before. It's really unusual for this part of the country to get that hot. Unfortunately, it was the exact same time when the cool season legumes, such as the lentils and the peas, were flowering in their regeneration plots. And a lot of regenerations failed that year. They either didn't make seeds or they didn't grow, even if they were irrigated. And on the right-hand side of the screen, it shows, based on the application results, that going forward, we're going to see higher temperatures more and more frequently in the Pullman location. Here's an example affecting the prunus collection. The prunus collection has been experiencing warmer, shorter winters. It's in Davis, California, again on the West Coast, but farther south. This is an example where we have sweet cherry accessions that aren't achieving their full dormancy in the wintertime. And as a result, their phenology isn't the way we expect it come springtime and summertime. We have delayed pollination and foliation and flowering, and we have reduced yield and performance. And if we look at the map going forward, we're going to continue to have warm temperatures in the coldest quarter of the year, and we need to be seriously thinking about what to do about this collection. And as a result, we actually have a new project going on where we are grafting part of the prunus collection in upstate New York and planting new trees there. And we're hoping that the trees will survive the winters there and will have deeper dormancy for those trees. And then I can cryopreserve them successfully, back them up, and they'll be safe in a place where they perhaps will grow back. We keep a pecan collection in College Station, Texas. This is in the very south of the United States, in the center. And there's been a lot of high temperatures. We have drought that's increasing a disease called shuck decline. And this results in poorly filled nuts and premature shuck split. We also have a problem with high temperatures during pollination. And the breeding program there especially had issues because inside their bags, the temperature was getting over 40 degrees Celsius, and many of the pollinations failed. And again, if we look forward, the temperature of the warmest quarter in the future is going to continue to go up. Climate change is also causing warmer temperatures and increased precipitation in the tropics. And we have 3 GenBank locations in the tropics in the USDA system. We have Hawaii, we have Miami, Florida, and we have one in Puerto Rico. We have a new collection of coffee that we're establishing in the Hawaii location. And unfortunately, coffee leaf rust is spreading to that place. So more things to worry about and more issues for pathologists. Pollinators, the insects that we depend on during a regeneration effort, also have a reduced efficiency in high temperatures. And these are some examples of the pollinators that we use inside those cages to make sure we have successful pollination events. And unfortunately, we can also have more pests with high temperatures. There are reports that European corn borer, black cutworm, coffee leafworm, and others will all be more problematic in American crops, as well as possibly in the GenBank going forward. As part of our planning effort for climate change issues for the National Plant GenBank System, we have developed this ebook that I mentioned earlier. And the last chapter of this ebook lists a number of resources that people in the United States and anywhere in the world can use if they want to do some adaptation planning. It gives examples of other reports that people have written for climate change adaptation planning. And it gives some guidelines and some references and some links for workbooks and resources that have been developed by other institutions. So what's the National Plant Germplasm System going to do? Well, we need to do a lot, and we're just now starting these plans. One thing is that we need to monitor the responses to environmental changes in our collections. And maybe we'll even have some materials in our collections that are doing really well. And this could be valuable for GenBanks or for breeding programs or for selection for new crops. When we do plant evaluations, we should be testing the plants under a wide range of conditions. If we just grow them out under the current conditions, we don't know how they're going to perform going forward. We need to do a really thorough assessment of the crop locations where the plants are growing now and see if they meet our current and future needs. We should be considering alternative power sources because we'll probably have higher greenhouse and cold room cooling costs in the future as we try to maintain conditions that the plants can live in. We really should have weather stations at each of our sites, and we need to back up or duplicate our clonal collections. We also need to back up our seed collections, but they're in better shape in general than the clonal collections are. Part of my job responsibilities are to back up these clonal collections, and so I'm going to talk a little bit about that next. I'm located at the National Laboratory for Genetic Resources Preservation in Fort Collins, Colorado. We have about 82% or 500,000 accessions backed up in our building, either at -18 freezer storage or in liquid nitrogen, stored as seeds. However, we only have about 15% or about 5,000 accessions of clonally propagated materials at our facility. This is because they're a lot harder to store and much more expensive to process. And sometimes we don't even have methods to store them. And we've got to put them in liquid nitrogen. We store things primarily as shoot tips and dormant buds, but we also have some pollen. Here's an example of clonal collections in our GenBank system, where we have citrus in the field in California. We have strawberries in a greenhouse. We have garlic in cold storage between seasons, and then it gets planted every year. We have in vitro collections, and then we have everything backed up as much as we can in liquid nitrogen. This is just a montage of all the different crops I have the opportunity to work with. I'm really lucky that I get to work across a lot of different crops and collaborate with many different people. We work on developing and implementing cryopreservation systems for shoot tips and also for dormant buds in my project. I just have a couple slides about each of these different techniques to tell you about them. First, we have dormant buds. These are materials that are, these are branches harvested off trees, the small twigs, the last year's growth, in the middle of winter. And for our temperate crops, they go really dormant in the wintertime. And you can see a picture of an apple bud on the upper left-hand side that we would harvest from the trees that are growing in New York State. They send these twigs to us in Fort Collins, and we cut them into single node sections, a couple centimeters long each. And then we dry them at 0 degrees Celsius, so in the cold, to about a 30% moisture content. Now we have dry little twigs that we package into tubes with 10 per tube. And then we put them into cans, and then we place them in a slow cooler. The cooler is dropped in temperature to -30 degrees Celsius at 1 degree Celsius per hour. And then it sits there overnight. And from there, we can take these tubes and directly put them into liquid nitrogen, and they'll survive. We can pull them out of liquid nitrogen by warming them for a couple weeks in moist peat moss in the refrigerator, and then they get grafted onto rootstocks to see if they're alive. And on the right-hand side, a viability test shows a sprouted bud growing on a rootstock in a greenhouse. We have a huge collection of apple accessions cryopreserved this way. It's over 2,400 in our cryotanks that we've been using this method for over 30 years. We also cryopreserve things using shoot tips. These are taken from either greenhouse materials or tissue culture materials, and we excise out teeny tiny 1 millimeter meristems with a few leaves. They are treated with cryoprotectants, which remove the water and replace it with things like dimethyl sulfoxide and ethylene glycol and sucrose, which allows them to be exposed to liquid nitrogen successfully. We needed to remove the water because if the ice crystals are still, if the water is still in there, ice will form when it gets exposed to liquid nitrogen. We then plunge it into liquid nitrogen using this plant vitrification solution 2, shown on the right. And it's cooled really fast. Once we get it into liquid nitrogen, we can pull it out of liquid nitrogen and then test its viability to make sure the little plants can still grow. And they are plated onto media. We can use this for almost any species if we have the method and the tissue culture all figured out. Especially useful for things that are tropical or that don't have dormant buds. So the dormant bud system is not an option. However, there's much more labor involved. Here's an example of where we use it for grapes. So we introduce shoot tips from grape plants in our National GenBank, which has over 4,000 accessions, into tissue culture. Then we make nodal sections and we excise off the shoot tips, treat them with cryoprotectants, put them into liquid nitrogen, package them into vials, and put them into long-term storage. We can revive out 1 or 2 vials to check the viability, and you can see them growing on the right-hand side. This project was actually established early on in collaboration with the Egyptian National GenBank, where I had a collaboration with Dr. Hassan and even hosted one of her students. It's an example of grape accessions that were successfully cryopreserved using the method we developed. And we currently have 40 accessions that we've successfully cryopreserved. Next, I have a slide about citrus. Citrus is really important to agriculture all over the world. And in the United States, there's a disease, citrus greening, that's really, really bad. And it became a priority about 10 years ago to back up our citrus collection so it wouldn't be lost to this disease. So we developed a method. We harvest these growing shoots from the materials in the trees that are growing in pots inside a greenhouse in California. We surface sterilize them, excise a shoot tip, treat it with cryoprotectants, and then plunge it into liquid nitrogen. Then the trick for citrus is that when it's time to recover it, we have to micrograft those 1 millimeter shoot tips onto seedling rootstocks that we started in tissue culture. So we grow some seedling rootstocks in the dark. We make a little ledge. We place a shoot tip on the ledge, and then we allow it to grow in tissue culture. Here's an example of it growing 2 weeks after liquid nitrogen exposure, then 4 weeks, then 8 weeks, it's rooted, and we can move it out to the greenhouse. We have over 450 accessions of citrus cryopreserved at our building using this method. And here's just a big list of all the types of crops we work with. I gave you a few examples, but we work with many, many more than that, both developing the methods and implementing them, making us one of the biggest cryopreserved collections of clonal materials in the world. The last part of my presentation, I want to talk about a new project we have. And this is one where we're developing training materials for plant genetic resources management, or gene banking, and use. And we developed this project back in 2018 is when we started planning it, because there's a lot of people retiring and there's a lot of turnover. And we realized that people didn't have the skills they needed to be gene bank managers in the United States. And we wanted to build a resource that anyone in the world could use. So we held some meetings and we came up with a plan. And our plan is to develop this website that's public. Anyone can use anything on this website. It's called grin-u.org. It's called GRIN because that's the National GenBank data system we have. So that's how you learn about our GenBank is by going to GRIN, to the GRIN database online. So we made one branch of this database be all about educational resources called grin-u. And anyone can access this and anyone can subscribe just by clicking the box on the right-hand side. If you subscribe, you get a quarterly newsletter telling about the new releases on our website. For this project, we've been writing a lot of different ebooks, and these ebooks are fully public. They are full of chapters with information like videos and pictures and downloadable PDFs and references and text. And you're welcome to use this content in classes or for training or for on-the-job training or for anything else. In fact, some of the ebook chapters that we have in this in our resources were developed during COVID when we couldn't come into the lab to do research as part of a grant project we had with the Egyptian GenBank. So instead, we created some ebook chapters for training and sharing information. The most popular ebook we have is kind of amazing to me, is about clonal cryopreservation. It's the one on the upper right-hand side, where we talk about all the different methods and that we use in our lab to cryopreserve shoot tips and dormant buds. But we have lots of other material in the form of ebooks also, and we're constantly writing more of them. Here's an example of the citrus ebook, where you can, or the ebook about clonal cryopreservation and the citrus chapter, where you can see the chapter, see the authors, and then watch these videos of members of my staff doing all the steps in the procedure. Another one we're excited about is we're writing a success story book because we want to tell the world how plant genetic resources and gene banks make a difference and how it's important to have our plant genetic resources secure, whether it's in the USDA gene bank or in any gene bank. So we're compiling success stories and sharing that information. The YouTube channel. As part of this project, we developed a grin.u.education YouTube channel that has a fair number of subscribers, and it currently has 74 videos. So anyone can access this and use our videos. The most popular video we have is in the lower right here. It's about the Native Americans and how they used corn and squash and beans together for companion planting. And we've got over 200,000 views. We also have virtual tours. So we made these 3 virtual tours about conserving genetic diversity, about seed storage, and about apple cryopreservation, about our facility. And anyone can look at these videos and see them. We make infographics. And again, through my collaboration with the Egyptian GenBank, we have them even available in Arabic. So these are downloadable infographics about plant genetic resources and their use in breeding, as well as some general information about gene banks. And you can go to the website and download them, print them out as posters, or even use them as one-page handouts. We have a couple more, one about the National Plant Germplasm System and one about botanic gardens and their role in conservation. And those are just available in English, Spanish, and one's in French. The last thing I'll mention is a class we're teaching. We developed 3 one-credit classes, which are 5 weeks each, that we offer online. This is in collaboration with my colleague at Colorado State University, Dr. Jeff Morris. And he teaches the first class for 5 weeks online that is about plant genetic diversity and measuring it, and the origins of plants and crop wild relatives. I teach the second 5-week class. It's about gene bank conservation. And then Jeff teaches the third class about using plant genetic resources in breeding programs, as well as genotyping and phenotyping. Information about these 3 classes that you can take 1, 2, or 3 of in any order. And they cost a couple $100 each because they're offered through the university, and they can give credit or non-credit options, and they're asynchronous. So people from all over the world take these classes, and they participate in discussion groups and share information, and they can watch the videos on their own. So Jeff and I are involved in the discussion groups, but anybody can watch the required videos that week, and they don't have to watch it on US time because we know we're often in a different time zone than the rest of the world. So with that, I would like to thank a number of my collaborators. I have many, many of them all over the world and within the United States. I feel very fortunate. I wanted to show a couple special pictures here from when my collaborator from the Egyptian GenBank, Dr. Hassan, visited us in Colorado, as well as when I visited the National GenBank a few years ago in Egypt. And with that, I'm happy to take any questions. Thank you for your attention.
Thank you, Dr. Volk, for the nice presentation. And now we open the discussion. So you can write your question in the chat or just raise your hand and I will unmute your mic. So do we have questions? Okay, I may start, Dr. Volk. So do you think that a cryopreserve methods can also fit with cereals crop like wheat and barley and legumes crop, or just it is specific for specific crops?
You can definitely, well, okay, the grains and legumes can be stored for a really long time. In fact, many of the grains are predicted to be able to be stored 1,000 years just in the freezer conditions. So you can store your seeds really well in the freezer, and there's no need to have cryopreservation for those seeds because they're safe as they are.
Okay.
The reason why we use cryopreservation for some things, such as even intermediate seeds that have a really high oil content and don't store well, or for shoot tips and dormant buds, is because we can't put them in the freezer and it's not an option.
Okay. We have like a question from Ahmed Haggag. I will unmute your mic.
Oh.
Yeah, please unmute your mic and tell your question, please.
Hi, hello, Volk.
Hello, good to hear from you.
Yeah, me too. Thank you.
You visited us in Fort Collins.
How are you?
I'm good. How are you?
I'm good. Thanks for your great lecture. It was very amazing. And actually, I want to from you send us the link for the new website that you created. I think.
Gladly.
Okay. So I didn't hear about it before, so I want to check it. Okay. And if it's okay, you can send us the lecture that you already give it to us right now. Okay, it will help us. And thanks for your great amazing lecture that you gave to us at 2018 as I remembered. I used of some of its slides when I was at a visit of for Lebanon. I gave it to some Lebanon students. Okay, and it was very amazing to them as I remembered. And actually, I want to discuss some points about the thermotherapy followed by cryotherapy, something like this. You didn't mention something of this in the lecture. So I do not know if you did some cryotherapy recently at your work. Did you meet some work as a cryotherapy?
Yes, I did not mention that because I didn't think I had time.
Oh,
but
yeah.
We've been working on cryotherapy. Cryotherapy is when you use cryopreservation methods in the shoot tip system, and viruses and viroids can be eliminated. And so it's an application of cryopreservation for pathology problems. And we published a paper last year, actually, on apple shoot tips, where we eliminated 2 viruses and 1 viroid. And it was really exciting because we didn't have another way to remove these diseases from our apple plants. And in fact, now our national quarantine system is building a cryotherapy part because they think this is going to be important for some other diseases that they can't remove using heat therapy or shoot tip therapy or other systems that pathologists use. So it's really exciting. We have several review papers on the topic, and this apple paper we just published last year.
Yeah, okay. Okay, I just want to know if you did some work on cryotherapy recently or not.
Yes, we continue to. We don't have any training materials about it yet, but
Yeah,
people keep asking me. So I need to write the training ebook chapter. It's on my to-do list.
thank you.
Feel free to email more with me, and I can share references.
Okay, thank you. Thanks much
Thank
for
you.
your
Good to hear from you.
Thank you.
Yeah, we have Dr. Volk another question in the chat. So thanks very much. I ask about how we can conserve pollen grain for olive plant or date palm.
Oh, what a great question.
Yeah.
I've actually cryopreserved all of the pollen from the date palm in our national gene bank. We have about 40 accessions, and we have them all preserved as pollen. Date palm preserves really, really well. I'm going to put my email in the chat, and you are welcome to email me, and I can send you references about how we do that. I'm also writing an ebook about pollen cryopreservation, and it's not quite finished yet, but that actually has a whole chapter about date palm cryo. I haven't tried olive yet, but I think it would work really well also. We've done cryopreservation of pollen for date palm, for prunus, for pecans, and for walnuts so far. And I'm sure we could do more, but it takes a very close collaboration with the people at the locations to give us pollen at the perfect stage so that we can process it successfully. So feel free to email me, and I can send you a lot of information about date palm. We've published multiple papers about it. It's actually very easy and successful, and it lasts a long time.
Okay. Okay, do we have a questions or? Yeah, please ask your questions or by raising your hand or just writing in chat room. Okay, I think we don't have further questions. Okay. So at the end, I would like to thank you again for presenting your outstanding research and experiences as well in preservation of genetic resources, important genetic resources. And I would like to thank attendees of this lecture. And this lecture is recorded, so it may be soon available at our YouTube channel, maybe at the end of August, after we finish exam work or. And so thanks for all, and thank you again, Dr. Volk, for your presentation, and hope to see you again through MGBS lectures.
Thank you very much for this opportunity. It's been really exciting. I appreciate the invitation.
Yeah, thank you. Okay, have a nice day.
Okay,
Bye-bye.
thanks. Bye-bye.